Welcome, everybody. For this week's lecture, we're going to be talking about voluntary standards, regulatory compliance, and all the non-technical issues that dictate how a biomaterial gets from the design stage to market. Because up until now, we've talked about the bulk, the surface properties, what makes a material biocompatible, and how we actually test for that, and what the FDA wants to see as far as testing goes. Now we're finally seeing what the FDA process is like and how we actually bring a design to fruition. So let's get into it. For the first few slides, we'll focus in on what makes an idea successful, or in general, what gives it value. And I really like this quote that the book gives: "An invention by itself is of little value. The value of an idea represented by an invention is through innovation, which is to say, the validation and implementation of that idea or embodiment thereof to affect individuals and populations through its utilization and acceptance by others." And the meaning of this is really embodied in the first part of that sentence, that an invention or an idea by themselves are not very valuable. I really learned this through a few of the companies that I've worked for over the years, where you might get a patent on something, but the patent itself isn't really that valuable. It might become valuable later because it represents a legal controlling interest in a particular innovation, but unless you show that that idea has some sort of backing, unless you establish its credentials, and unless you show how valuable that particular idea is, it's really just a legal tool used to hold a controlling interest in that idea. However, once you patent an idea and then you start bringing it through all those biocompatibility tests that we've covered early in the lectures, once you start bringing it through in vivo studies, the preclinical setting, and you start establishing how important that idea could potentially be, only then will people start taking interest in your particular device. Now, medical devices, and certainly companies that develop medical devices, have a number of hurdles to go through before they can establish value to their particular design or idea. They include fulfilling complex regulatory requirements. They need to obtain reimbursement, which is to say that they need to get medical insurance companies on board to potentially reimbursing patients or to offer payment for their products so that patients don't have to pay out of pocket for it. And then finally, even on top of all that, they have to be appealing to any one group of medical practitioners. The last part is to say that you can show both safety and efficacy during the regulatory trial process, but you could still somehow be more expensive, you can take more time, you can be more cumbersome or less convenient to medical practitioners. So in that way, they would never use your device. In describing some of the criteria that devices have to meet in order to be approved or in order to be successful, I've also kind of started to describe some of the ways that devices and companies can potentially fail. So in LVMR, you might have an uncompelling need. So if you can't justify and if you can't sell your device, even if it's been proven safe and shown efficacy during the trial process, it's not going to be picked up. The market size perhaps doesn't justify the cost to develop the product. We certainly see this with orphan drugs where there absolutely is a compelling need, just not the market to justify the costly process to bring something through clinical trials. Also, an idea could potentially infringe on other patents. Sometimes it's not even sure if they do infringe or not, but just the potential to go through legal proceedings is enough to thwart an idea from coming to fruition. Lastly, there could be the case where insurance companies don't offer reimbursement for your particular device, drug, or idea. Therefore, patients having to pay out of pocket for it likely won't, because devices are expensive, and thus your product could be on the market and it could be FDA approved, but you won't have buyers for it. Now, when bringing a device to market, there are a few factors that are considered important for success and a few others that are less so or not important at all. Some of the important factors include a proven decrease in mortality or morbidity, which means to show a clear and significant efficacy in your particular product. You may be able to show a substantial decrease in cost. Your particular device or drug might filter out unresponsive patients, so those that fail to show any benefit from it, or even the lack of side effects or adverse effects of your particular device or drug. Lastly, you might come up with some sort of diagnostic method that leads to earlier diagnosis, and most importantly, that early diagnosis leads to a better outcome. This is in contrast with the less or not important factors where you might have an early detection, but that early detection leads to no benefit to the patient. Perhaps surprisingly, the improvement in patient comfort is not a guarantee for success, particularly when the outcome remains unchanged for whatever device or therapeutic that is being developed. And then lastly, any slight decreases in surgery time are less of a factor for success than you might think as well. In part because initiating surgery is such a difficult part in itself that if you're only shaving off a few minutes from that surgery time, it's just not worth it to bring that device to market because that in itself is not going to be a compelling reason for a surgeon to use that particular product. Next up, we're going to cover this flow diagram that details the stages of a particular device or drug from conception to adoption and widespread use. Something I also like to call the circle of life for biomedical engineers. And it all starts with, intuitively, the new idea, the stage that was creatively called in cerebro. But this is the stage where you come up with a concept, and now you want to move on to the next stage to see if it works, which is the proof of concept. Now, what distinguishes the proof of concept to later stages is that the audience for this is simply yourself, i.e. the engineer, and any other supporting members of your group or team or company. Meaning people that are already invested in this particular project, people that are certainly biased and want it to work, and it's really just a stage to see, okay, is this something that I should be spending my time on? And the book has a great quote on this. It says, "One dumb experiment is worth 10 expert series." So really, it's the first demonstration to yourself and insiders that your idea that you came up in the previous step is worth pursuing. Now, this is in contrast to the next stage, which is the proof of feasibility, in which case the audience are skeptical outsiders. These might be, for instance, investors that are prudent with their money, who understand how medical devices or drugs in general fail, and thus want to kind of poke holes at any type of demonstration or result just to verify that something isn't going to pop up later down the line after they've invested their money. The next stage ties the theoretical functioning of the product, which you've already demonstrated in the previous stages, to the practical requirements for using it. And all things considered, it's really just an exploration of whether or not the product can add value. Some factors included in this are the time to perform a particular procedure or to implement the device, the expertise required, practice pattern, patient comfort, maybe time for rehabilitation, and the likelihood for any adverse outcomes are all considered at this stage. Now included in all the previous steps are various iterations of in vitro, in vivo, in situ testing, and now which brings us to the first point, which is the first human use. This is a really monumental step because it requires a lot of hurdles to go to. It means your company has already managed to persuade an institutional review board, an IRB, to allow for the device to be used in patients. It's received an investigational device exemption from the FDA, which allows for testing in humans. And these all require significant preclinical testing to show some form of safety, even in small idealized patients in the first clinical stage. Now note that this stage is simply getting something into humans. It has nothing to show for safety and efficacy in humans, which is the next step, clinical utility. And this is really just early statistical confirmation of what has so far been kind of anecdotal evidence or benchtop testing to show that your product may have value. The big difficulty with this stage is that it takes a lot of time to develop. A lot of devices, especially biomaterial ones, are ones that require large patients to use them and extensive time for them to be used. The next stage is when some sort of regulatory body, convinced by the previous stage in the clinical utility, grants approval to market that particular device. We'll cover this in a little bit more detail in later parts of this lecture. The next stage involves getting people to actually adopt your particular device. It's only been approved so far, but so far, beyond the clinical trials, no one's using it. And only 5 to 10% of clinicians are what would be characterized as early adopters, meaning ones to readily invest in a new technology. Mainstream users, on the other hand, need a lot more to justify using a new type of technology, and particularly getting training in to use something like that. It would require testimonials from early adopters, discussions at medical conferences, and journal articles to convince them to move over to a new standard of care. And that's exactly what the next stage is. When your product has now received widespread use, it represents a significant portion of the market at this point, and it's also having large competition from knockoffs and incremental or substantial improvements from their previous device made by competitors. And it's really at this part where the deficiencies or the need for the improvement are very readily apparent, and that's where we come full circle to the new ideas that bring about new innovation for the next device or drug. So now you can see why I call this the circle of life for biomedical engineers. Next up, we're going to talk about the regulatory approval process a little bit more in detail. And one thing I want to highlight up front is that there's no unified way that a device gets approved or cleared. We've already highlighted a number of differences between drugs and devices. So in fact, the FDA is split up into 3 different divisions to differentiate and undergo different approval processes depending on what exactly your idea constitutes. Those 3 divisions are first the Center for Drug Evaluation and Research, which handles obviously new drugs. This division, by the way, has the most difficult and stringent standards to get something approved. The next division is the Center for Devices and Radiological Health, so this is where typically a biomaterial will go through. And lastly, there would be a Center for Biologics Evaluation and Research, which is where vaccines, live biotherapeutics like probiotics, cell, tissue, and gene therapies would go through. However, monoclonal antibodies, despite being a biologic, are regulated by the Center for Drug Evaluation and Research. Now, it's certainly possible to understand that there are many combination products that might be both a device and a drug, a device and a biologic, or perhaps even all 3. So, of course, the first thing that the company and the FDA want to understand is who's actually going to regulate this. Because it certainly might be the case where a biomaterial device that has a drug component to it would be regulated by the Center for Drug Evaluation and Research as opposed to the Center for Devices and Radiological Health, which would itself carry more difficult and stringent standards. The companies for this case would file what's known as a request for designation, which is often made to determine the classification or to make an argument for where that classification and regulation should lie. This is absolutely what a company like Cordis, a subsidiary of J&J, would do when they developed their Cypher stent. This was a combinational product that had both a device and drug component to it, and because of the more stringent standards of the drug branch of the FDA, Cordis wrote in their request for designation to the FDA that the, quote, primary mode of action is to provide a mechanical buttress that resists mechanical compression. The coating on the stent serves the ancillary purpose of retarding formation of intimal hyperplasia. So that language, primary and ancillary, they're very much making their argument for the less stringent devices, and they were granted that, and the Cypher stent went through the Center for Devices and Radiological Health as opposed to the Drug Evaluation and Research branch. Now, within the devices branch of the FDA, there are different classification of devices based on the type of risk that they pose to a patient. Furthermore, many products are cleared to be sold based entirely on equivalence with already cleared devices. And in order to do so, companies have to demonstrate that their product has a substantial equivalence to an already cleared product. In order to do so, that device has to have the same intended use, the same technology characteristic, or different technological characteristics that do not raise any new questions on safety or effectiveness. Now, the traditional classifications split devices into 3 separate categories, the first one being Class 1 devices, which pose the lowest risk to patients. Now, currently there are 782 different types of Class 1 devices. In order to be marketed as any of these type of classes, by the way, you have to fall under a certain designation of a type. And if it's a brand new type, it's likely going to be in the Class 3 until proven otherwise. And there might be half a dozen or so different types of gloves or different bandages, for instance. And somewhere on the FDA's website, there are appendices with all the different types and class designations so that you can simply fill out the paperwork and designate what type you are. Good examples for Class 1 devices are, again, bandages, gloves, adjustable beds, some surgical instruments, stethoscopes. And I have gloves bolded here simply to remind me of a story that might illustrate the loose standards for Class 1 devices. There used to be gloves that had powder coatings to them that were made out of like talc material so that whenever your hands sweat, you could very easily take out these gloves and put on a new one because that absorbent powder had already absorbed the sweat. They're very handy for cell culture work, radiological work, where you very routinely took off many gloves and put them on throughout the day. But back in 2014 or 2015, the FDA stopped classifying this particular device to be cleared for Class 1 approval, for the Class 1 designation, in part because there was a growing amount of evidence that's saying that talcum powder was linked to cancer. Now, this wasn't a large recall. They weren't banned from use. Companies were still allowed to sell them as long as they still had the inventory. Hospitals were still allowed to use them if they had them on hand. It simply meant that if you want to market a new type of glove that you had It had to fall under a different type classification that didn't have talc included in that. And part of those requirements are listed under what we consider general controls. These controls include the device listing, the labeling of the product, and then the FDA quality system regulations. These QSRs are in place so that manufacturers must establish and follow quality systems to help ensure that their products consistently meet applicable requirements and specifications for that particular device's type and class. In short, you can usually bring any of these devices to market without prior approval, and you can start manufacturing and selling them right away, as long as at some point you're willing to have the FDA perhaps conduct an audit of your particular facilities to make sure that those QSRs are in compliance. Class 2 devices pose a little bit more risk than Class 1 devices. As of a year ago, there are about 799 different types of Class 2 devices. Some common examples of which are electronic stethoscopes, catheters, needles, monitoring devices, surgical drapes. I think we can imagine in all these situations that if any of these products somehow fail to work somehow, we can absolutely start seeing potential adverse effects or just in general bad situations. These devices will be subject to the same general controls listed for the Class 1 devices, in addition to what are known as special controls, which include things like performance standards, post-market surveillance, and then certain FDA requirements that are specific to the different types of devices within the Class 2 classification. Class 2 devices are brought to market using what's known as the pre-market approval notification process, which is called 510K because that's the clause in the Medical Device Act that allows for this type of approval, or clearance, I should say. Now, Class 2 devices may require some sort of clinical or human studies, particularly if they lack any predicates, but this is nowhere near as rigorous as the next classification, which are Class 3 devices. Class 3 devices pose the highest risk, and by default, any new type or any new device is automatically a Class 3 device unless they can prove that it's somehow safer and should be designated to a lower class. Aside from some relatively less invasive devices like catheters or needles, almost anything that goes inside the body is a Class 3 device. If a device somehow doesn't go inside the body but is also a life-supporting or sustaining device or necessary for those applications, it's also going to be a Class 3 device. These devices will absolutely need clinical studies to demonstrate both safety and effectiveness, and they require what's known as the pre-market approval, PMA, to bring a new device to market. There are also two other less commonly used classification systems, the first one of which is the de novo classification system, which is reserved for new types that would by default otherwise be classified as a Class 3 device, but whose safety profile and technology are reasonably well understood, so much so that they shouldn't have to go through the rigorous standards of a Class 3 device. I just pulled up one random example here that I thought would be a good representative of the de novo classification system. This is the Sentinel Cerebral Protection System, which is based on technology very similar to the guidewire system that puts a stent in place. However, this is a device that actually used that guidewire to capture an actual thrombus or blood clot debris while performing transcatheter aortic valve replacement procedures and actually physically pulls out that embolism. Now, because no other device had ever been built for this type of purpose, it should have been a Class 3 device. However, the company was able to argue that because it's based on a prior technology just used in a different circumstance and thus would have a different label, they successfully argued that it would be a de novo classification system subject only to general and special controls because it had a reasonable assurance of safety. The other classification is called the Humanitarian Device Exemption, which is reserved for devices for orphan diseases or conditions, in which case there is less than 4,000 cases in the U.S. In order to get this classification, a company must provide evidence that there is no other way that this device could be brought to market and no comparable devices to prove that substantial equivalence on predicates are available. Again, one example that I happened to pull up was called the Deep Brain Stimulation, or DBS, which was approved in 2009 as a humanitarian device exemption for people with severe OCD. It's composed of transmitters that are implanted into the brain that act as sort of brain pacemakers, which is how the company describes them. Now, because of the large gap in effort required to clear a device as a Class 2 device through the 510K pre-market notification process and a Class 3 device, which requires the pre-market approval process, I want to talk about these two processes a little bit more in detail, and particularly to contrast the differences between the two. In 2009, ooh, these stats are a little dated. In 2009, the review fee for a 510K process was just under $4,000. It was $3,700. Whereas the same year, the review fee for the PMA was $200,000. Now, note, these are not the cost that it takes to conduct the trials. They're simply the filing of the paperwork and the follow up work that's required by the FDA in order to clear or approve these. And so use these numbers as an indication of how many man hours are spent approving or clearing these type of processes. Between filing and approval, it takes approximately 95 days for the 510K process, whereas it takes 335 days for the PMA process. Because of the costs and stringencies involved in the PMA process, it's very desirable to go through the 510K process, obviously. So a couple of these numbers really highlight this. In 2012, there was about 3,200 510K applications that were cleared, whereas in that same year, there was only 32 PMA approvals. 510K process generally does require laboratory data, but does not, for any specific rule, require clinical studies. They may be required, and about 10 to 50% of applications do require some clinical data, usually only to prove the parts of the technology that lack certain substantial equivalence or lack a predicate device to base them off of. So because these devices can show substantial equivalence, that portion of the technology that is equivalent has already been proven. So that in itself doesn't require human or clinical data. PMA process, however, does require substantial amounts of data, certainly from the preclinical, in vitro, and in vivo stage, but absolutely human clinical data. And you may have noticed I've been trying to be very clear about my language. Anything that goes through the 510K pre-market notification process is quote unquote cleared, not approved. The word approved is only reserved for PMA devices. So now the general PMA approval process, in part we've described, I think, about 80% of this throughout the lecture so far. There's the pre-submission process where you come up with an idea, you do your proof of concept and feasibility through in vitro and in vivo methods. Then you apply for what's called a investigational device exemption, which actually allows you to put a device into humans just to obtain data. That test has to be approved for by an institutional review board, which I'm going to explain in just a few minutes. Then, after you believe all the necessary trials have been conducted, you file for the PMA, which is a request to market approval by the FDA. Even after a product's been approved, there might be reasons to change a particular product. Maybe there's a new indication for use of that same device. Maybe the labeling has changed. There's been changes in the sterilization procedures, packaging, or the source of some of the raw materials. So then a company might have to file what's called a PMA supplement. That would allow the FDA to then approve those changes. Now, an IRB is an in-house group of people, meaning that they will be employees of the company. Universities have IRBs to approve studies conducted by researchers. And the purpose of the IRB is to ensure that all research is conducted with appropriate safeguards for human subjects. So if it involves humans, an IRB has to be approved. There's an equivalent IACUC board that's used for animal studies. For humans, it's IRBs. IRBs must have 5 members, one of whom has an interest that is primarily scientific, and then one member whose interests are primarily non-scientific. The focus of an IRB includes many things primarily related to the safety of the patient. So there's certainly a risk-benefit analysis that occurs using the same chemical and hazard classifications that we learned about in one of our previous lectures. The board is tasked with ensuring that patient confidentiality is maintained and that the patients are informed of the process that they're going through and that they consent to it. They're also tasked with ensuring that appropriate selection of subjects have been chosen so that the experiment can include whatever it is that the researchers need to prove. Also, many of these clinical trials are incentivized so that patients sign up for them. So there's even an evaluation of the incentives that are given to patients and making sure that the patients perhaps aren't bribed and is somehow revealing a good result. And then lastly, there's also plans for a continuing review of the data as it comes in, even after the study has been concluded. I also want to know that the IRB is actually tasked with deciding whether or not an IDE is required or not. So once they do the risk-benefit analysis that I mentioned first, they actually decide if a significant enough risk is imposed, in which case the company has to file for an IDE. If there's no risk involved at all, then the FDA doesn't require that. But that's the IRB's decision, not the researchers or the FDA. So now I'd like to get into a few legal case studies that I think help illustrate the type of liabilities that a manufacturer, that engineers, and that patients and medical practitioners can fall themselves under. The first one involves pedicle screws, which are bone screws that are implanted into the pedicles of the spine to anchor stabilizing hardware during surgery. Now, in 1993, there was a 2020 expose that publicized the off label usage of these screws in other types of bones. This culminated in a lawsuit titled Buckman versus Plaintiffs Legal Committee, a class action lawsuit. And the lawsuit alleged that the company committed what's called fraud on the FDA by failing to disclose the studies that they had conducted internally on the off label usage of these screws. Furthermore, the lawsuit contended that the manufacturers were liable because they failed to disclose of the data they accumulated from these off label studies and disclose of the inherent risks to patients. Now, the court threw out the fraud charge because, in the end, the court was okay with the non disclosure of studies for off label usage. In these cases, the manufacturers weren't trying to market these screws for off label usage, so for only their intended use, they turned over all the relative studies. The court furthermore felt that disclosing everything related to all these screws would just be too cumbersome for the FDA, and thus only required the relevant data to be disclosed. This case also describes what's called the learned intermediary doctrine, which states that manufacturers have a duty to warn medical practitioners and the medical community at large, but not the patient, in part because it's up to the medical community and these clinicians to understand the dangers, where it's not reasonable to understand that the population at large would. A few other case studies deal with artificial heart valves. Now, artificial heart valves have gone through a few different designs over the years, the first of which involved a ball and cage valve that would fail due to major embolic complications and catastrophic ball escape, where these balls would actually escape from their cage. And then, I'm imagining this sort of cue ball sized ball going down an artery or a valve. There's also improved models that are called leaf designs that have these leaves that kind of like flutter open and closed, that could potentially suffer from potential and catastrophic fatal failures from the strut fractures. So in this case, the leaves would actually come undone, and then they would travel downstream as well. So there's a big major court case involving artificial heart valves called Bowling versus Pfizer, where Pfizer is forced to pay $75 million in a patient benefit fund, $80 to $130 million in a medical compensation fund, and then a further $10 million for spouses of class members. What's notable about these cases is that there are a few other separate lawsuits that occurred successfully, meaning that the plaintiffs were awarded money, that occurred without actual valve failure. The first example being Kahn versus Shiley. Now, this is where a lawsuit where a defendant, after hearing about the Bowling versus Pfizer cases, sued the defendant because the patient had already received this heart valve that had been not recalled by the FDA, but certainly a lot of these adverse events were showing up. And what the lawsuit, interestingly enough, resulted in is this sort of stipulation that the manufacturer, in these kind of cases, had the burden of proof where they needed to show that their particular device, in this case a heart valve, was not in the process of failing. And because they failed to do so, they had to pay the plaintiff money. In another case, Michael versus Shiley, a patient suffered quote-unquote tangible injury because they had to go through surgery to remove this heart valve that had in itself failed, but because of the risk of it failing was so catastrophic, the patient wanted it out and replaced with another device, and they successfully sued the manufacturer because of the sort of risk that they had to go through and the tangible injury that resulted from surgery. And now a monumental court case that I want to discuss a little bit more in detail is Regal versus Medtronic. That has to do with the patient Charles Regal that was undergoing an angioplasty procedure when his catheter burst and caused him permanent disabilities. Charles Regal then sued Medtronic. The case ended up going all the way to Supreme Court, arguing negligence on the design, manufacture, and labeling of the device. Now, the Supreme Court found in an 8-1 decision that the state tort laws that Charles Regal was suing under were termed strict liability, which normally only have to prove that something went wrong. They don't have to prove any sort of mindset like that would be required in fraud or negligence. They found that to be, quote, different from or in addition to, unquote, federal FDA regulations and thus invalid. And what these are rooted under is the preemption clause in the 1976 Medical Device Act that prevents litigation under common tort laws. Now, I really like this quote from Justice Scalia. It says, "The PMA process is a rigorous process. The FDA spends an average of 1,200 hours reviewing each application and grants pre-market approval only if it finds that there is a, quote, reasonable assurance, unquote, of the device's safety and effectiveness." Now, in short, the PMA approval is in no sense an exemption to federal safety review. It is federal safety review. And in short, what this prevents is to have an uninformed jury making decisions on safetynesses of devices when in fact already informed experts in the scientific and medical community have already done so through the PMA process. A second equally monumental case that also was argued before the Supreme Court was Wyeth versus Levine, which also has to do with the preemption clause. Now, this particular case has to do with promethazine, a first-generation antihistamine and sedative that was used as an anti-nausea drug and marketed by the pharmaceutical company Wyeth. Now, one particular patient receiving this drug, Diana Levine, was given an improper IV dose of this drug that resulted in gangrene and required amputation of her arm. Ms. Levine sued Wyeth and under state liability laws was awarded $6 million in damages from Wyeth. Wyeth then sought to recover those damages, particularly in light of the new preemption clause case study of Regal versus Medtronic, and sued all the way up to the Supreme Court. Supreme Court, however, in contrast to their previous decision, Regal versus Medtronic, found in favor of the plaintiff and concluded that the FDA approval of drugs does not preempt state tort law. In an opinion for the majority, Justice Stevens wrote that, quote, "Wyeth's failure is based on the misunderstanding that the FDA, rather than the manufacturer, bears primarily responsibility for the drug labeling. It is the central premise of the FDA that the manufacturer bears responsibilities for the contents of its label." So, while both drugs and medical devices go through rigorous safety trials, it has to do with how they communicate its use and their potential adverse effects that have to do with their labeling and why one might be covered by the preemption clause and exempt from state tort laws, whereas others aren't. Now that we're finally touching on drugs and pharmaceuticals, I want to touch a little bit on their approval process. Now, any new drug has to go through the aptly named New Drug Application Process, or NDA, which is the equivalent of the PMA process for devices, and then the Biological License Application, BLA, which we did discuss, for any biologics in terms of rigorous and standards. Within an NDA, a company needs to establish a few things. The first and foremost is safety. So do the benefits outweigh the risks for that particular drug and for that disease? They also need to show efficacy, which is the capacity for sufficient therapeutic effect or beneficial changes in a clinical setting. They also need to show labeling. So what's this indication of use, any warnings and precautions, and then doses necessary for the particular patient. And then they also need to demonstrate sufficient standards in their manufacturing process. In part, what makes the NDA process so rigorous is that there is no equivalent Class 1 or Class 2 510K clearance process like there are for devices. So we want to think of every new drug as almost the equivalent of a Class 3 device that needs to go through the PMA process. And then even on top of that, NDAs are themselves more rigorous than PMAs in terms of patients needed, the time it takes, the cost, and a few other factors. Just like new devices need to go through an IDE, investigational device exemption, in order to start gathering data during the clinical trial stage, drugs need to go through a investigational new drug application, which is whenever there is a new indication of use, a change for the approved route or dosage, changes in the patient population that that drug is targeting, or significant changes in the promotion of the approved drug. Now, there are a few exceptions to an NDA process that, while they do require rigorous clinical data, might somehow accelerate the approval process. Perhaps in the same way, these might be similar to the de novo or humanitarian exemption for devices. A few of them are fast track designation, accelerated approval, they give priority review, or they might be considered a breakthrough therapies. So a lot of these drugs would be intended to treat some sort of serious or life-threatening condition. They might answer some sort of unmet medical need, so they might be the first in their kind type of classification. And they also might have the significant improvement of safety or efficacy over existing therapies. So the idea is that the FDA would want to get these out as quickly as possible because they somehow pose a greater benefit to the population than some other minor, say, headache medication, for example. So a lot of these designations, like fast track, accelerated approval, priority review, imply some sort of accelerated approval of that particular drug. So I'm not going to get into the specifics that differentiate them, but a lot of the common benefits to these is that they somehow get some expedited review. They might have what are called surrogate endpoints, where instead of relying on a disease to go all the way to, like, for instance, cancer, we don't have to wait for a patient to be particularly cured, but there might be some sort of accelerated approval endpoint where a tumor gets reduced at a shorter time frame so that a drug can be approved faster without having to wait through the entire prognosis of a particular disease. And there might also be what's called a rolling review, where a drug, particularly for orphan diseases, might be granted approval on limited data. And then as real world data comes in and more patients start using a particular drug, the FDA can continue to monitor it and make a final approval or disapproval after enough sufficient data has been collected that would otherwise have been difficult to obtain during the clinical process. Now, the drug approval process can be significantly more lengthy than a device process, so I want to highlight all the different phases that drugs have to go through. There's still a preclinical step, in which case there's in vitro studies, in vivo studies, any R&D required before humans are introduced. There's potentially a phase 0 phase, which is often skipped, the goal of which is simply look at pharmacokinetic testing as opposed to safety and efficacy. So in this case, you would have a very small subtherapeutic dose in a limited amount of people. This would only occur over the form of months, just to track the pharmacokinetics and metabolic breakdown of the particular drug. These can often help solidify what type of doses are required for the actual phase 1 through 3 trials. The goal of phase 1 testing is simply safety. In this case, we're using healthy volunteers, not even those with a particular disease, because we want to look at any adverse effects that might occur or toxicity with this particular drug. You would still give a therapeutic dose, a subtherapeutic dose, sorry, and the participants will only be ranging in the 20 to 100 of, again, healthy volunteers. And this might occur over 6 months or so. During this time, usually only 70% of drugs make it through the phase 1 process. A big change in phase 2 is no longer using healthy volunteers, because now we want to start gathering, in addition to the primary goal of assessing safety, we want to start getting a little efficacy data as well. So we want to start testing this in patients with that particular disease for that drug is intended to treat. So in this case, you would also give a therapeutic dose, so elevating the dosage that are given to patients. And you also start expanding the sample sizes of these studies, anywhere from 100 to 300 patients. And this might take anywhere from 1 to 2 years or so. And 33% of the drugs that go through phase 2 make it onwards to phase 3. Now, the goal of phase 3 is to test everything. We want to look at efficacy, effectiveness, and of course, all safety. So safety is a recurring theme in all of these. Again, we're dosing at therapeutic levels, and we're certainly dosing to a larger sample size of patients, into the thousands now. Up to 3,000 patients perhaps. And this might take up to 4 years or so. And usually if you made it this far, only 25% of drugs make it past phase 3, which is usually approval. You can also go into a specialized phase 4, which is a post-approval process where you're gathering real-world data. So phase 4 is a very formalized version of that. Certainly drugs that make it past phase 3 that aren't required to go through phase 4, manufacturers still like to gather real-world data in just less clinically relevant settings. Now, there are a number of things that really aren't working in the favor of drugs in terms of the clinical trials and what takes them so long to conduct. One reason is that chronic conditions like cancer just take months and years to see if a treatment has any effect. Very often, patients simply aren't willing to undertake the time commitments necessary to continue on with treatments. In fact, 85% of clinical trials fail to retain enough patients for their particular study. Furthermore, drugs with minor side effects simply require larger sampling sizes than normal in order to determine statistical relevance for both safety and effectiveness. Now, it can be so hard to actually get and recruit patients that about 40% of the entire total pharmaceutical clinical trial budgets go entirely towards recruitment. And as one can imagine, only certain patients with target diseases are eligible to participate in certain clinical trials. And this requires physicians who treat disease to participate in the study, identifying suitable candidates for that study, and then also obtaining consent to use a new drug in those particular patients. And as patients get tired of going through the rigors of a particular study, certainly a lot of them drop out. In fact, 30% of patients drop out of clinical trials. Others can unfortunately die during treatment. So, from these numbers, we can start imagining that in order to get the necessary 3,000 patients to finish a clinical trial, how many patients initially needed to be enrolled in that particular study in order to finally dwindle down to that final value of 3,000. It certainly might be more than 10,000 patients would be necessary. Now, I'm going to end the lecture on that. I've included a few supplemental slides that, if you have time to, you can review. I think they give great figures and data on the size of clinical trials and the difficulty of going through them and the costs associated with them. And then in class, we'll talk about some of these differences a little bit more in depth. So, thanks for listening.